Thank you so much for the opportunity for me to present. On behalf of my co-investigators, I'm pleased to present the clinical activity of the RET inhibitor Pralsetinib, also known as BLU-667, in patients with RET fusion positive solid tumors. RET alterations have been characterized as oncogenic drivers in multiple cancers. RET fusions are seen in 1 to 2% of non-small cell lung cancer, 10 to 20% of papillary thyroid cancer, and RET mutations are seen in over 50% of medullary thyroid cancer in the sporadic type, and almost 100% of patients with germline as a part of MEN2 syndrome harbor these RET mutations. Beyond non-small cell lung cancer and thyroid cancer, we also see RET fusions as oncogenic drivers across multiple disease types, although they are extremely rare. For instance, we see RET alterations in cholangiocarcinoma, colorectal cancer, ovarian epithelial cancer, breast cancer, and including tough-to-treat epithelial tumor types like pancreatic adenocarcinoma. Standard therapies provide limited benefit for patients with RET fusion positive tumors. Outcomes with immunotherapies, unfortunately, that we know that with patients with RET fusion positive non-small cell lung cancer are also poor. Genome-driven precision oncology, as we know, has altered the landscape of multiple hematologic and solid tumor malignancies. Recent tumor-agnostic drug approvals, such as pembrolizumab across all mismatch repair defect diseases, and larotrectinib and entrectinib across all NTRK fusion positive solid tumors, have resulted in a paradigm shift in cancer treatment away from organ histology-specific indication to a biomarker-guided targeted approach. Pralsetinib, also known as BLU-667, is a potent and selective RET inhibitor that targets RET alterations, including fusions and mutations, regardless of the tissue of origin. Pralsetinib, in the preclinical studies, is specific and has high selectivity for activating RET alterations and has very low activity for other kinases. So the ARROW study is a phase 1/2 clinical trial with this agent Pralsetinib that had multiple ascending doses of this drug, and the phase 2 dose was reached. And the dose was 400 milligram once a day convenient dosing. From this, a phase 2 study was designed with 3 tumor types. So what are the eligibility criteria for this study? Advanced solid tumors, the patient need to have RET alterations with local TXT. And the primary endpoints were centrally reviewed objective response rate per RECIST version 1.1 and safety. 3 cohorts were there as a part of the ARROW study. The first cohort was RET fusion positive non-small cell lung cancer. The second cohort was RET mutation positive medullary thyroid cancer. And the third cohort was RET fusion positive other solid tumors. The RET fusion positive non-small cell lung cancer, the overall response rate or the objective response rate in treatment naive patients was 73% in RET fusion positive non-small cell lung cancer, and 61% in post-platinum patients with RET that harbor RET fusion positivity, 61% overall response rates. In the RET mutation cohort, mainly consisting of the medullary thyroid cancer, the treatment naive objective response rate was 74% in RET mutated medullary thyroid cancer and 60% in those previously treated with a multikinase inhibitor like cabozantinib or vandatinib. This study is going to be mainly focusing upon the all-comer solid tumor basket cohort, which is the RET fusion positive other solid tumors. Again, what are the baseline characteristics of these patients? So 13 of these patients had RET fusion positive thyroid cancer, and 14 patients had RET fusion positive other solid tumor types. Again, these RET fusion positive tumor types are difficult to treat epithelial cancers like pancreatic cancer, cholangiocarcinoma, thymic cancer, colon cancer. And the RET fusion positive thyroid cancer cohort consisted of papillary thyroid cancer, and all of them were radioactive iodine refractory patients, and 1 patient with poorly differentiated thyroid cancer. Let me go ahead with showing the activity of Pralsetinib in RET fusion positive thyroid cancer. So the disease control rate among all RET fusion positive thyroid tumors was 100%. The objective response rate was 91% and the partial response was 91%. Again, we showed that this was active across all fusion among all the RET fusion positive thyroid cancers. The partial responses and the responses were rapid within the first 2 or 3 months, even in patients that were progressing on radioactive iodine. And in this time, let me also share a patient that I treated with RET fusion positive thyroid cancer who had a poorly differentiated thyroid cancer. As we know, these are very difficult to treat thyroid cancers. And on cfDNA liquid biopsy testing, we showed that the patient's tumor harbored the RET fusion. This was later confirmed in the tumor tissue. The patient, when he presented to our clinic, could not breathe because the tumor engulfed the esophagus and trachea. A tracheal stent had to be put in so that he could breathe. Because of this RET fusion, he was enrolled on this Pralsetinib study. And within 2 to 3 weeks, the patient started feeling better. The infiltrating mass surrounding and compressing the thoracic trachea decreased in size. And eventually, throughout treatment, the patient had a deep and durable partial response with Pralsetinib of 18 months duration. In fact, the patient had 94% shrinkage of the target lesions. The thyroglobulin antibodies also reduced from over 1,400 to less than 1. It also showed concurrent complete clearance of the RET fusion ctDNA in the liquid biopsy. So let me go to the activity of Pralsetinib in other difficult to treat epithelial tumor types. The disease control rate was 92%. The objective response rate was 50%. The partial responses were 50%, and the stable disease was 42%. Responses were observed in all patients with pancreatic adenocarcinoma, 3 out of 3 patients, and cholangiocarcinoma, 2 out of 2 patients. Again, many of these patients who were enrolled on the study had only FISH testing done. Again, the main thing is that we don't know if the FISH was a false positive fusion because the specific partner is unknown. And we also showed that Pralsetinib was active in RET fusion positive solid tumors, irrespective of the tumor type, including cholangiocarcinoma. Again, I'd like to share an anecdote of a patient of mine with cholangiocarcinoma who his tumor harbored the RET fusion. Again, when the patient was sent to our clinical trials early phase drug development clinic, the patient had progressed from all standard lines of therapy, including gemcitabine, cisplatin, and Abraxane, which is nab-paclitaxel. The patient's tumor harbored the EGFR activating mutation, for which the patient was on erlotinib, bevacizumab, and subsequently with osimertinib. And the patient had progressed on all that. Patient, when she was sent to clinic, was in a wheelchair bound, and her performance status was rapidly declining. Given that her tumor harbored this NCOA4 RET fusion, we enrolled the patient in this clinical trial. Within 2 to 3 weeks, the patient started feeling better, and she could get up from the wheelchair and she could start to walk. And her tumor markers, which consisted of the CA99, reduced from 1 million to 82 throughout treatment. Also showed concurrent rapid clearance of the RET fusion ctDNA in the liquid biopsy. In fact, the patient, from being wheelchair bound and unable to perform the activities of daily living, could go on a ski trip to Europe within the first 4 months of starting treatment. This undoubtedly conferred clinical benefit and gave the gift of time to this 51-year-old mom of teenagers. Again, these are the responses that we see in these patients that give us more motivation to do early phase drug development with these novel therapeutic targets. What about the safety? The safety among the 27 patients with other RET fusion positive solid tumors is consistent with the safety of the overall population, which is being separately presented at the ASCO meeting, poster number 9515. What were the major adverse events? The most common treatment-related adverse event was greater than 15% were anemia in 1/3 of patients, and increased AST, which is a liver function test, in also over 1/3 of patients, decreased white cell count in another 1/3 of patients, hypertension in 30% of patients, increased ALT in 26% of patients, hyperphosphatemia in 19% of patients, and neutropenia in 19% of patients. The treatment duration was up to 23.5 months with a median relative dose intensity of 96%. In fact, in this all RET fusion positive solid tumor cohort, the basket cohort, no patients discontinued treatment due to treatment-related AEs. In summary, Pralsetinib has broad and durable anti-tumor activity with multiple advanced solid tumor types, with a 91% objective response rate and 100% disease control rate in RET fusion positive thyroid cancer, and 50% objective response rate and 92% disease control rate across various RET fusion positive solid tumors, including responses in these multiply relapsed and heavily pretreated patients with hard-to-treat epithelial cancers like cholangiocarcinoma, pancreatic cancers, and neuroendocrine tumors. Again, my personal pitch is these are rare tumors. And these tumors were found mainly based on genetic testing. So the more we test, the more we find. Again, the key message here is to keep looking for these rare RET mutations and rare RET fusions. In summary, Pralsetinib, also known as BLU-667, was well tolerated, and its safety profile was consistent across the overall population treated. The ARROW study is still ongoing and currently continues to enroll patients with various RET fusion positive solid tumors. I'd like to thank all the patients and their very motivated families who traveled very long distances to enroll on this clinical trial. We also thank the Pralsetinib investigators globally across both sides of the Atlantic, in Asia, everywhere in the world, and colleagues at Blueprints Medicines Corporation, and all the clinical trial personnel and research staff who enabled, even at the era of COVID-19, working hard remotely or on-site so that these patients could have access to these life-saving therapies.